Hello dear students, बहुत सारे दिन हो गए मुलाकात नहीं हुई after so many days. I was actually very busy in some specific tasks at college, so that is why I could not upload a video. I was missing all of you, and I was thinking of getting some time to upload a video for you people. Today I'm uploading a video for you people, and hopefully inshallah I'll try to upload at least one video every day so that my students can be with me and they can be up to date to some extent in the concepts that I am trying to cover. I cannot teach anybody anything. I can only make them think this is what I am trying to do. I will give you the concepts and you people will learn, and you will learn the concepts and learn how to apply the concepts in different fields of life. Okay. Now, first of all, let me go to the share screen and then let us see what we are going to study today. Today, we will be studying a new topic, and this is for class first year. The new topic that we will be studying is the topic of enzymes. Enzymes are actually catalysts. Catalysts, all of you know, they are those factors or those chemicals that tend to speed up a chemical reaction. But because we are going to talk about the chemical reactions that are taking place in living bodies, which include bacteria, which include fungi, which include plants, which include animals, any living thing in which the reactions are going to be speeded up, that is going to be known as a catalyst. But because this catalyst is present in the living body, we will call it a biological catalyst. We will call it a biological catalyst. And specifically, we will call it an enzyme. It will be called an enzyme. And the chemical reactions that are taking place in the bodies of living things, be it bacteria, be it fungi, be it protists, be it any living thing, animals and plants, whatever, you name it, and it is a living thing. These chemical reactions, they are said to be metabolic reactions. Metabolic reactions, as all of you know, metabolic reactions are actually of two types. They are of two types. We have the building up reactions, which are known as anabolic reactions. A very good example of building up reactions are photosynthesis. Photosynthesis is a building up reaction. The formation of glycogen from glucose in the liver of humans is a building up reaction. It is an anabolic reaction. And then another type of metabolic reaction is the catabolic reaction. The catabolic reaction is the breakdown of substances from larger molecules into smaller molecules. For example, respiration. In respiration, larger molecules are broken down, and they are broken down after combining with oxygen, and then they produce carbon dioxide and some other byproducts, water, etc., etc. Okay, that is a catabolic reaction. Digestion is a catabolic reaction. Starch is broken down by amylase into glucose. Proteins are broken down into amino acids. Fats are broken down into the combining members, and that is the glycerol and the fatty acids. So these are all catabolic. All these reactions that are taking place in the body of living things, they are speeded up by special types of factors which are called enzymes. Enzymes are biocatalysts, and their function is to speed up the reaction, right? This is the introduction of enzymes. I will take you to the presentation also, but first of all, let me give you a general overview of what the enzyme is. Now, enzymes, how do enzymes speed up reactions, metabolic reactions? How do enzymes speed up metabolic reactions? How do enzymes speed up? There is something in a combining two combining substances. They contain some amount of energy, and that amount of energy has to be overcome for the reaction to take place. And that energy is known as the activation energy. That is known as the activation energy. That activation energy is the barrier that the reactants have to overcome to carry on the reaction. Enzymes, what do? They lower the activation energy. Enzymes lower the activation energy and make it easier for the reactants to combine together. Now, let me draw a graph so that you people can understand. It won't be very clear, but I'll try to draw the graph for you people over here. Now, if, for example, let me draw the axis of the graph. Okay, this is the axis of the graph. You can say X and Y axis of the graph. Okay, X axis here, and this is the Y axis. I'm just going to tell you what activation energy is. That is why I have drawn this. Okay, right here. Now, this is the energy level that is required for the products, for the reactants to acquire before the reaction can take place. Yeah, this is the energy required. Now, this energy required is actually the activation energy. The reactants have to acquire this amount of energy before the reaction can take place. And what do enzymes do? When we add on enzymes, enzymes tend to lower the activation energy. Enzymes tend to lower the activation, and they lower the activation energy to this extent. Now, look here. This is the extent to which the enzymes have lowered the activation. And let me show you the difference now. This is the reduction. This peak has been reduced. This is the lowering of the activation energy. This is what enzymes do. They lower the energy required for the reactants to combine. Here we have the reactants, here we have the products. This is the x-axis, this is the y-axis, right? This was just to explain to you people. Okay, now let us go on. Enzymes have specific types of characteristics. Enzymes have specific types of special factors. Now, first of all, what is the nature of enzymes? Most of the enzymes are protein in nature. I said most of the enzymes, not all the enzymes. Most of the enzymes are proteins in nature. And what type of proteins? They are globular proteins. Globular protein means they are soluble in water. They are soluble in the liquid medium. They are globular proteins. We have proteins which are fibrous proteins. Fibrous proteins are those proteins that are, for example, they are present in our bones, in our hair, in our skin, in the cell membrane. We have fibrous proteins. Fibrous proteins are insoluble. Globular proteins are soluble. All our enzymes are globular proteins in nature. And what level of organization are they? Most of them are in the tertiary level of organization. They are in the tertiary level of organization, most of the enzymes. Now, most of our enzymes, I have told you, they are protein in nature, but we have another group of enzymes which is called the ribozymes. Ribozymes are those enzymes which are something except proteins. They are made up of RNA. And that is why we say that ribozymes are made up of nucleic acids. Ribozymes are nucleic acid enzymes. And a very good example of ribozyme is peptide transferase. Peptide transferase is an enzyme that is very, very active during the amino acid converting into during the process of translation. The formation of the polypeptide bond is because of this peptide transferase enzyme. And this peptide transferase enzyme is in the group of ribozymes. It is not a protein enzyme. This peptide transferase is present on top of the ribosomes. And it is the enzyme that is responsible for the formation of the peptide linkage between the incoming amino acids. Right now, let us come to some of the characteristics of enzymes, right? Some of the characteristics of enzymes. First of all, when we look at the structure of the enzyme, the structure of the enzyme is made up of two main parts. Two main parts. That is the body and the active site. Okay, the body and the active site. The active site is that area where the reactants are going to combine. The active site is divided into two parts, the catalytic site and the binding site. Active site is divided into the catalytic and the binding site. It is divided into the catalytic and the binding site. First of all, the substrate attaches to the binding site and then it comes to the catalytic site. And then the catalytic site, it works. This active site actually is a specific distribution. It is a specific ionic distribution, or you can say ionic arrangement, or you can say arrangement of amino acids. This active site, it is a specific ionic distribution or specific amino acid combination. These amino acids, they come together. I have already told you that enzymes are tertiary in nature. These amino acids or these ions, they are not necessarily present on the same part. They might be present on far away regions, but after the folding, they come into a three-dimensional region. This three-dimensional region is known as the active site. And this is where we have the binding site by which the products, the substrate is going to attach, and then the catalytic site where the reaction is going to take place. One more thing you have to keep in mind that enzymes, this was the first characteristic. Secondly, enzymes are used in a very small amount because enzymes are not destroyed during the chemical reaction that they are catalyzing. Enzymes are not destroyed, they are preserved. They can be reused and reused and reused again and again, right? So we do not need a very large amount of enzymes, right? After that, we see that enzymes are very, very specific in nature. For example, amylase is an enzyme that will always digest starch. Protease is a group of enzymes that will digest only proteins. Give amylase 2 molecules of starch and give amylase 20,000 molecules of protein. Amylase is never going to digest protein. It will always digest starch because it is very, very specific. All enzymes are specific. However, we have some enzymes that have 2 or more substrates. A very good example of such an enzyme is ribulose bisphosphate carboxylase, commonly known as Rubisco. Rubisco is an enzyme that is very commonly seen in the Calvin cycle. This Rubisco is seen in the Calvin cycle. Ribulose bisphosphate carboxylase. In the Calvin cycle, Rubisco the substrate for Rubisco is carbon dioxide. But in other reactions in the body, the substrate for Rubisco is going to be oxygen. It has an attachment, it has a specificity for carbon dioxide and for oxygen, both at the same time. In this condition, in the Calvin cycle, it acts as carboxylase. And in some other reaction in plants, it acts as an oxygenase. So it has a specificity for oxygen and carbon dioxide, both of them. Then we have another site, another characteristic of enzyme, and that one is that enzymes, they show a very specific structure, and this structure is known as the binding site. The binding site is actually it is very specific in shape. If the binding site is destroyed, then the catalytic activity of the enzymes, it gets destroyed. Okay, you can get a question in that what is the structure of the active site? What is the structure of the active site of enzymes? What is the structure of the active site of enzymes? You have to explain this by saying that it has a specific ionic distribution. These might be present far away from each other, but when the amino acid is folded, they come together into a 3D position. Now, enzymes, they are there's another characteristic. Enzymes, they are very specific to heat. They are very specific to heat. They are sensitive to heat and pH. Enzymes can work very well in vivo. In vivo means inside living bodies, and they can also work in vitro, in laboratory situations, in glassware. But the situation is such only when you give them the specific conditions. It will work in vivo. God has created the conditions in our body. It will work in vitro. In glassware, in the laboratory setup, when you give it the specific heat and the specific pH conditions that are required by the enzyme, except that enzyme will not be able to work in vitro. Okay, so you have to keep this in mind. Let me go back to the presentation and let us see what we have in the presentation. Okay, now I will share another screen with you people, and we will go to the presentation and see what we have in the presentation. And if you want, you can note down the presentation. And then after this, the next topic that we will have is cofactors and prosthetic groups. Right, introduction. The enzymes are actually molecular substances that are present inside. Enzymes are globular substances or molecules with catalytic properties. Until recently, it was thought that all the biological catalysts are enzymes, but now it is not so. This is the enzyme structure. I just told you the question that write a note on the active site. Enzymes are complex three-dimensional globular proteins. They are not straight proteins. You know that proteins has three structures. I'm going to upload a lecture about the structure of proteins also very soon. Some might have other associated molecules also. Only a small part of the enzyme is very active, which is going to carry out the reaction, and that is the active site. And it actually comes in contact with the substrate. Now, this is the whole protein arrangement of amino acids. Now, these three regions, they are present far away on the protein polypeptide chain, but the protein is folded in such a manner in this tertiary structure that they come close together and they produce this active site. And this active site is going to be very suitable for the attachment of the specific substrate, which is going to carry out the reaction. Amino acid chain folded into its own enzyme configuration. And this is a very specific configuration. Properties of enzymes. They are proteins most of the time, and the activity can be easily affected by temperature and pH. Enzyme by temperature denatures them. pH dismantles the ionic distribution. Enzyme increases the rate of reaction by lowering the activation energy barrier. I've already told you this, thus allowing reactions to proceed without an input of energy. Enzyme, how does it react? It forms an enzyme substrate complex to facilitate the interaction of the reaction. I'll explain this point on the board right now. An enzyme only changes the rate at which the equilibrium is reached. It does not change the ratio of the equilibrium, not at all. A small quantity of the enzyme is needed for the reaction because it remains unchanged throughout the reaction. Enzymes are very specific for their substrate. Some enzymes will act only on a particular substrate, other will act on similar molecules. I have just told you the example of ribulose bisphosphate carboxylase. It acts as a carboxylase, it acts as an oxygenase. Many will break a particular linkage. Example, hydrogen bonds. Enzymes break down particular linkages. For example, when starch has to be digested, the glycosidic linkage in the starch is going to be broken down. When protein has to be digested, the peptide linkage is going to be broken down. When lipids have to be digested, the ester linkage is going to be broken down. So this is how enzymes break down the components. Enzymes do not alter the equilibrium of a reaction, but the speed at which it is reached is increased. Now, this is another enzyme, carbonic anhydrase, which is very important in the chloride shift reaction, the transfer of carbon dioxide through the body. I have already discussed this in one of my previous lectures. I have discussed this process in very, very detail. You can go and look at the video if you want. Now, this is how the activation energy is going to be reduced. This is the free energy of the substrate. It has to acquire this energy barrier before the reaction takes place. So products are always at a very low level. Enzymes reduce the activation energy and make it easier for them to carry on the reaction. Enzymes are antibodies with catalytic properties, and ribozymes are molecules of RNA which act catalytically on themselves. I have told you example of peptide transferase. Right? This was all for the lecture today. I hope you enjoyed the lecture. Next lecture will be about cofactors and coenzymes. Stay tuned for the next lecture. See you people next time. Okay, take care of yourself. Goodbye. Allah Hafiz.